the wonderful opportunity to conduct research in the Balko lab, where I investigated how VU809, an inhibitor of MYC and WDR5 interactions, impacts macrophage differentiation and polarization. So a little bit about me. I was born in Raleigh, North Carolina, and I currently attend North Carolina Central University, where I'm majoring in biomedical sciences and minoring in Spanish. I've always had a passion to go into the medical field, inspired by my mom, who is a breast cancer survivor. Seeing how doctors were there for us during that difficult time, I hope to be there for patients and their families in the future. I've also had the wonderful opportunity to conduct research during undergrad, and I really like how you're able to improve treatments in the bench that can potentially be used in the clinic someday. In my free time, I enjoy playing the violin, doing arts and crafts, traveling the world, exploring nature, and playing badminton. So in the Balko lab, we study breast cancer. It's the most common cancer in the US, where 1 in 8 women in the US will develop breast cancer in their lifetime. It is also the second leading cause of women cancer deaths, emphasizing the importance of having improved treatments. One thing that makes breast cancer difficult to treat is the complexity of the breast tumor microenvironment. Here, tumors send signals that rewire immune cells that prevent immune cells from promoting immunity and instead allow progression of the tumor. An immune cell that is found here is the macrophage. So you may be wondering, what are macrophages? So they are cells of the immune system involved in phagocytosis. So you can imagine like the Pac-Man eating all the bad stuff, the bad bacteria. And then it's also involved in wound healing. So there is a variety of macrophages and they span a spectrum. One end of the spectrum is the M1, which are pro-inflammatory or tumor inhibitory, and on the other end are M2, or tumor-associated macrophages, which are anti-inflammatory and tumor supporting. So my project this summer involves looking at differentiation and polarization. So here you can see differentiation is from the precursor monocyte to macrophages, while polarization is from macrophages looking at the specialization of the macrophage, so like into either the M1 or the M2. So macrophages play roles in cancer. There is the tumor-associated macrophages that I mentioned before, which play roles in angiogenesis, cancer progression, and immunosuppression. Changing gears, another area of interest is the transcription factor, MYC, because it regulates effectors that play roles in cell proliferation, differentiation, and immune surveillance, as well as several other roles displayed. MYC promotes polarization of M2-like macrophages, which can lead to immune evasion, according to literature. So recruitment of MYC to chromatin is dependent on interactions with WDR5, shown here. This can promote gene expression, leading to proliferation, which can lead to cancer. We are focusing on a small molecule inhibitor, VU809. VU809 binds to WDR5, which can lead to loss of WDR5 from the chromatin, decreasing transcript levels of WDR5 bound gene. So our question is, does inhibiting MYC and WDR5 interactions alter macrophage differentiation and polarization using VU809? And so our hypothesis is, if we have macrophages, we add the drug, then we expect higher expression of M1 markers such as INOS and lower expression of M2 markers such as arginase 1. So prior to looking at differentiation and polarization of macrophages, we wanted to validate the activity of VU809 in our hands. So we used tumor cells and then conducted 2 different assays. First, looking at the cell cycle progression as well as the target engagement of the drug. So looking at cell cycle progression, you can see here, when we add the drug, there is less cells in S phase. This supports that VU809 decreases cell cycle progression. Looking at real-time quantitative PCR data, we expect that when we add the drug, there will be less expression of WDR5 responsive genes. And this is actually what we see here. When we add the drug, there is less expression of RPS24, WDR5 responsive gene, and a slight increase in the WDR5 non-responsive. So first, we differentiated the bone marrow cells into macrophages using this protocol here, and then plated with MCSF. Here is an image of the bone marrow derived macrophages that we used in the experiment. So our experimental design involved using mice bone marrow. Then we differentiated it into bone marrow derived macrophages, and we looked at it with and without the drug, and then proceeded to do flow cytometry to analyze macrophage differentiation. Next, we also did look at macrophage polarization. First, again, differentiating it into bone marrow derived macrophages, and then polarizing it into M0, M1, and M2 macrophages, using interferon gamma for M1 polarization and IL-4 for M2 polarization, with and without the drug. Then we analyzed for gene expression of M1 and M2 markers using real-time quantitative PCR. So according to the flow data, we can see that the percentage of CD68 positive cells increases from about 30% to around 12%, and decreases, and then the percentage of Ly6C positive cells increases from around 50 to around 80%. This supports that VU809 reduces differentiation into macrophages. According to the real-time quantitative PCR data, we looked for how the drug impacts polarization of macrophages, and we can see that when we add the drug, we can see that it increases M1 expression of INAS, and it decreases arginase 1 expression, which is an M2 marker. This supports how VU809 increases M1 and decreases M2 marker expression. So our conclusions are, when we add the drug, we see a reduction of differentiation, where we saw there was a decrease in the percentage of CD68 positive cells, which is a macrophage marker, and an increase in the percentage of Ly6C cells, that is immature myeloid cell marker. And that supports how VU809 reduces macrophage differentiation. Another conclusion that we found this summer is when we add the drug, we saw a higher expression of M1 marker, INAS, and a lower expression of M2 marker, arginase 1, supporting how VU809 impacts macrophage polarization. So in the future, we hope to examine the effect of the drug on macrophage phenotype in murine mammary tumors, as well as test if treatment with the drug improves the response to immunotherapy for breast cancer. So without further ado, I would like to thank my PI, Dr. Justin Balko, and my mentor, Dr. Hanna, as well as the Vanderbilt MSTP Summer Research Program and the Balko Lab, as well as several others. Thank you so much for listening, and I hope you learned something new. Any questions?